Everybody welcome. So today is our great pleasure to have Maxat Kulmanov, who will present their work on the called DeepGO and DeepGO-SE. So this is a special software to predict protein functions based on the semantic entailment. This is a very interesting method, and I am very interested to hear the details about this. Maxat, thank you very much that you agreed to make a talk, and please start.
Thank you, Alex. Thank you, Alexander, for kind invitation and introduction. It is my great pleasure to present our work here. And thank you for all the participants coming and, yes, devoting their time. So my name is Maxat Kulmanov, and I would like to present our latest version of DeepGO methods. I think this is our fourth version of DeepGO methods, and I will also maybe briefly talk about our previous versions here. So I come from a university called KAUST from Saudi Arabia, King Abdullah University of Science and Technology, and I work in a group called Bio-Ontology Research Group led by Professor Robert Hoehndorf. So our group, we've been working more than, it was established in around 2014. It's around 10 years since we have started. So this work was done in collaboration with people from Swiss Institute of Bioinformatics and University of Geneva. And yeah, let's get started. So yeah, first of all, I would like to introduce Gene Ontology and in general protein function prediction. So when we say we predict protein functions, we actually mean we predict gene ontology annotations, and gene ontology is a vocabulary, and it's an ontology that describes protein functions. It started around year 2000, and it has been developing since then, and it's I think the largest biomedical ontology, and it has a lot of axioms and a lot of knowledge in it. I think more than 100,000 axioms and more than 50,000 classes. And yeah, I'm sure you are very familiar and you know about gene ontology a lot. But anyway, so it has these three different sub-ontologies where biological processes that describe the processes to some which gene products, proteins contribute to, and the molecular functions, they are small functions that can be performed by a single protein or gene product. And the cellular component, which describes the locations where a gene is active and it's present. So yeah, this is what we mean by predicting protein functions. And the classes in this ontology, they are described using formal language, ontology language called OWL, and they are not independent. They have a lot of relations between each other, like different types of relations, and they stand like there are axioms and so on. So all this comes from the the definitions of ontology, right? So the reason gene ontology was used because the ontology was used to describe functions, because the protein functions are not easy to describe, and the ontology comes here as a tool to that can do this, can describe very complex knowledge, and it's defined as explicit specification of a conceptualization of a domain, and basically you can it's a set of axioms that are true in some domain, and it has mainly three different types of axioms in it. Those are like T-box axioms, the their class axioms, where you define classes and then the relations between them, and there are ABOX axioms, which you can use to describe the individual instances of these classes, right? You can assert, for example, in our experience, when we come for the function, we describe the functions using these axioms. There are other ways, of course, but like if this has functions, some Go and this individual protein P, for instance, for example. And also the relations can have axioms. The relations can stand in some other relation to other relations, and they are also can be described in RBox axioms, but there are very few of them in Go and in many other approaches. So yes, and in addition to the axioms, the ontologies, they have also another annotations that is also part of the knowledge. So they come with the labels. So they have, in all the classes, they have unique identifiers. They're like definitions that are described in using natural language, and labels also described using natural language. And then there are axiomatic definitions. For example, one class can be described using some other classes. For example, this cell activation involved in immune response will be like described using other classes like cell activation and then also immune response, subclass of axioms, and many other. So Go is very rich, and our main objective, our aim, our goal in these projects, in DeepGO projects, was mainly focused in the knowledge in the ontology. Okay, so we mainly in this work, in our last work, we had and also maybe in our previous work, we had our main focus was on how to efficiently and effectively incorporate this background knowledge in Go into protein function prediction model, because we believe that using this knowledge in Go, you could significantly improve the protein function prediction, because the labels, they have a lot of information. And another question we addressed in this the latest version was how to predict functions for uncharacterized proteins, because we it's if we look at many of the function prediction methods, many of them, they work very well for the well-studied proteins. So if this protein has a similar ortholog or some similar protein that is well annotated, so using the sequence similarity protein function prediction methods, they could assign the annotations easily, right? So, but our focus was to how would you predict for those proteins where you don't find any similar sequences, with annotations, with good annotations to transfer from. And so that was our setting in this last project. So I'd like to talk briefly about our previous method. So in the first version, we mainly wanted to address the first research question on how to incorporate gene ontology, the knowledge in Go, into our prediction model, and also how to learn some representations from sequence, right? So we came up with this model that uses CNNs, convolutional neural networks. At the time, they were successfully applied on images and many other type of information, and we applied it on sequence by representing sequence as trigrams, and then learning embeddings using these kernels. So we learned some representation using this 1D convolutional neural network. And the second type of information where we wanted to learn also was protein-protein interaction networks. Here we don't have the like model, but this embedding here comes from a method like called DeepGO-SE that we use to learn representation from the PPI graph for the protein. So, and if we did not, because protein-protein interactions, you don't get interactions for all the proteins. We just use the sequence similarity to identify the most similar protein and then use its interactions to learn this embedding. So we combine these two different type of information. And here in the classification part comes where we incorporated the knowledge in Go. So we took the hierarchical structure in Gene Ontology and we constructed this classification neural network where each node in this graph, each node would represent Go, and the structure of this graph would represent the hierarchical relations in Go. So if this is a specific class, it would also, the predictions of this specific class would go to the higher level, to the superclass. And then superclass also will use all the predictions from its subclass nodes. And this way we could, for example, consistently predict like without like fixing or without propagating the scores from lower levels to upper levels, because we know that like if a parent class has some prediction score, it cannot be lower than the children class predictions, because there's this true path rule that scores propagate to the top, and this model would like automatically do it also, and then use this information while training the data. So we have shown that this will like this improves the prediction results in some way when compared to the models that do not use this. However, yeah, we participated in CAFA with this model, and yeah, we did not perform like that well. I think in some evaluations we had good results, but in general, if we compare to other methods, like it was not very good. And yeah, because we had some limitations here, we could not predict, for example, all Go classes that were there because we needed training data. I think we only predicted out of this 50,000, we predicted only around 2,000 classes. And we did not use any sequence similarity based predictions here. So it performed well in our splitted evaluation, but when we came to CAFA time-based split, the methods that had similarity component in there, like they were outperforming our DeepGO. So yeah, in like in some other like after CAFA, like I tried testing this like and combining sequence similarity predictions, and results were improved. And then we wanted to like overcome these limitations, and like the aim was to maybe perform better in next CAFA. So we have developed this second model, DeepGO Plus, where we used a little bit different version of convolutional neural network. Instead of this embedding layer here, we used just one hot encoding, and then we had many different convolutional neural networks with different size of kernels, and maybe more like longer representation. We also removed the PPI part because many proteins, they were missing interactions, and we predicted we increased the number of classes. We removed also the Go hierarchy because it was not very scalable. It was very slow. And we wanted that to improve kind of protein function prediction performance over what we had. But we had to sacrifice some of the components to do this. Yeah, so and then this model, I think it gave better performance also in Kappa because it had sequence similarity part, and it was very fast. We could predict functions very easily, and we also had a website that can run predictions very fast in a fast way. But we used we still, like, I think after the prediction process, we fixed the prediction scores using the ontology structure. We using this true path rule. So there were some limitations here as well. So then our main focus was still the ontology, and we have been working on methods to learn from ontology structure to embed ontologies, and we have developed this model called EL embeddings. It did not predict protein functions. We did not test it at the time when we developed this. We only evaluated this model for predicting protein-protein interactions. But so the idea here was to learn and embed ontology into Rn, so represent classes in Rn, so in a distributed space, such that the properties of the ontologies, so the semantics of the symbols would be conserved, would be preserved in this Rn space. So, like, at the time, there were many knowledge graph embedding methods that were being developed, like trans-A and different versions of translational methods. So based on those, we came up with idea, because the difference between knowledge graph and the ontologies, that in the knowledge graph, you only have mostly the ABOCs, right? The individuals. So the nodes in the knowledge graph, they usually are like individuals, they're instances. But in the ontology, you have concepts. And you cannot represent a concept as a single point. So we, instead of a single point, we represented the classes in the ontology as an n-ball. So in two-dimensional, it will be a circle, where with a radius also. Okay. So the radius and then all the area of this circle would represent this class, and all the points in this that are in this circle would be instances of this class. So we used the trans-E property to model the relations between ontology classes, and we came up for EL ontologies that use the description logic EL, which we could normalize into 4 different forms of axioms, like C subclass of D or C subclass of exists RD. So, and these 4 normal forms, C and D is subclass of E, and exists R, C subclass D. So these 4 different types of axioms. We came up with 4 different loss functions. So the first loss function would go to 0 if, for example, if C is a subclass of D, then the circle of C, n-ball of C, should lie inside the n-ball of D. So in this sense, in that case, these loss function that we have developed would be 0. And when there's a relation, so this circle of D will be translated into another circle, and the C will lie inside. So this geometric model properties, we use the geometric model to learn an ontology model. So this was shown to improve protein-protein interaction task compared to other methods, and also knowledge graph embeddings. And yes, this was accepted in each guy. So the learning process, for example, if we take just a simple family domain ontology, so it would be like the first, in the beginning, all the embeddings and all the points will be 0, and then over time, when you learn, this geometric model will like be the same as the will preserve the structure of these axioms. For example, this male class will go inside the person, and then the father will be inside male, and so on. So when this all the no like constraints, all the constraints that we put using this axiom-based loss functions will be satisfied, then the training will stop, and then the loss will go to 0. So after this method, we wanted to incorporate and use this model to improve protein function prediction. And we came up with this next version of DeepGO, which was called DeepGO-SE. And this version of DeepGO is like was based on, it's like we removed this convolutional part, and in our experiments we found that if we just use the interpro scan based annotations, that is also like that also works and was even better in some evaluations. And we would use these features, interpro annotations, and an MLP model to project this protein into this space. Okay, and to predict the assertion axiom here, this has function go that class, and to predict, we came up with this formulation where we use the has function and then has function embedding. So this is the relation embedding, class embedding, and then the dot product of it to the protein plus its radius, and then the sigmoid activation function as a score, prediction score. And then we optimize this model jointly when we train the EL embedding, we end-to-end. So optimize the big loss function together with EL embedding loss functions together. So this model worked very well, and it also had some
I'm sorry,
additional.
about this.
Yes.
About previous slide. So do I understand correctly? So you train embeddings of the protein and ontology simultaneously?
Yes, together. So
okay, yeah, okay, thank you.
Simultaneously, yes. So everything would go to 0 together. So, and here we used all the axioms in the ontology and all classes, all 50,000 classes in Go, and all the axioms in Gene Ontology. Even if some of the Go functions, they didn't have any training instance proteins. Okay, and that gave us some improvements when we, for example, compared the specific annotations, the Go, very specific Go classes, and their predictions. We saw that their performance in general, for example, if you look in this graph, like the classes with like around 50 annotations, right? Their performance increased compared to a simple MLP model that didn't use any EL embedding loss. And also, this method is called DeepGO-SE because it allows us to predict functions of classes where you don't have any training protein. So there are many, like in Go, we have 50,000 classes, but only I think around 30,000 of them have some experimental annotations, experimental, like the proteins with experimental annotations. So you, like, it's using the standard ways, it's not possible to train any classifier for those functions. But here, since we have this background knowledge in gene ontology, and we know that some of the classes are related to each other, we can predict annotations even for them. So I forgot to include this figure here, but it's in, if you want the details, like, of the zero-shot predictions, like in the paper, there are more information here.
Maxat, there is a question also in the chat. So when you on the previous slide, when you plotted these circles, was it,
yes,
for gene ontology, was it a two-dimensional, or it is just a picture just to
no, this is not two-dimensional. It's
okay.
just like for the
Okay. Yes.
yeah, for the years of understanding, but the dimensions of in this model, I think we had dimension of embedding was around 1024
Okay.
here.
Okay. Thank you.
Yes, so this is just to visualize.
To visualize.
Yes.
And sorry, I have also a question. Is this zero-shot ability, does it come from the fact that you also optimize this ontology losses which you showed earlier, where you want, for example, the subclasses to be geometrically equivalent, that a subclass is then the smaller ball inside the bigger ball, or okay.
Yes, exactly. That's where it comes from. So because gene ontology has this knowledge, right? Some classes are defined using other classes, right? So if you have, for example, some proteins that are annotated, and you can learn these specific classes that are used in definitions of other classes, right? You already know these relations, and you can do the prediction without even like having training instance for that class with a definition, right? So in a similar way, like we use these relations and then the knowledge in Go to learn functions, to learn like associated proteins with them, even though there are no experimental evidence or annotated for them. So we tested this, like we removed we took some specific classes. We removed all their annotations from the training set, okay? But we knew that there are some annotations for them, but we removed completely from our training. We haven't seen a single protein that annotated to that class. And after the training process, we evaluated them, and we got, for some of the classes, we got AUCs of 0.9. It's in the paper, so I didn't include it here, but it's for some, it doesn't work for all of them, because some Go classes are not also well defined. But if there is knowledge about Go, and you can learn this relation between classes, then in that case it worked. Even, for example, if there will come some new class in the next version of Go, right? And it uses some definitions that are in the previous one, even like for those, you could use the definitions in order to come up with the prediction, because this model allows, like, if you have the definition for a Go class, you can even learn an embedding without even like retraining the model from scratch. You can just use the embeddings and then use these transient properties to learn an embedding for this class to compute an embedding for this class, and then you can use this embedding in somewhere here in the to do the prediction.
And maybe related to this also, do you take also the protein embedding which you get from some kind of for example from ESM or something like that, and project it into the same space? And
yes, that's
what's the effect of that, and why you do this?
And that's
I mean,
what,
to project.
That's what's coming here in the latest DeepGO-SE version, right? So in the DeepGO-SE version here, we are using ESM2 because we in our experiments, and also the second version of ESM, like we did this with the first version also. I'm not really sure, but at the time it didn't work well. Maybe there were some other problems, not the ESM one, but when ESM, the second model came, they also have shown that in their paper that it also learned it's good for predicting protein structures, because in ESM they have shown that their embeddings encode for protein, like this very far evolutionary relations of proteins, and even some structural information is encoded in this ESM because they have ESM fault, right, that predicts structures from this ESM2 embeddings. And we decided to use ESM and then project into this embedding space. That was one improvement here. What, like, we changed the interpretations to ESM. So this is the final DeepGO-SE model. And another thing we did is we modeled this prediction process. We changed it as a semantic entailment. So in logic, right, in description logics, the semantic entailment is in simple words, it's defined as if you can infer a knowledge based on the A box, so based on the instances and based on the models of the ontology. And if it's true in all models, then that means it's semantically entailed. So if you have like infinite models, and then your inference is true in all of them, that means that this this statement will be semantically entailed. Okay, so this is purely based on the individuals. So there are some, like, because we cannot train infinite models of this ontology, and some relaxations, like in the number of the true models and also in the number of the models. That's why it makes it approximate semantic entailment. So if this knowledge in our case, like if this knowledge in average true in average it's true in our models, then that we say that it's true in the prediction. So we combine, we train not only single, we train like independent models of geometric models of the ontology together with the embedding, and then we combine their predictions. We do the inference based on all of the models. So these models are just the different instances of the training. So there is no like maybe like we just use different random seeds, and however their prediction scores slightly differ in each training, and then you don't get the same model, right? And if our score, the average score, is true in these models, then we say that it's true. So we have some other ways of combining it, but when we use the average, this gave us the best results in our models. So yeah, this is the similar idea in DeepGO-0, but here we are using multiple models, and we are also using ESM2 embeddings to represent it. So, and we evaluated this model for predicting the functions of uncharacterized proteins. So the way we evaluated it, we used this hard split methodology where we split the proteins based on their similarity. So we compute the sequence similarity with diamond. And if two proteins are similar, we place them in a one group, and we make sure that our training and testing and validation set, they don't share any similar proteins. So if you use, for example, diamond and then try to find similar proteins in training, you won't find. So these methods that are based on sequence similarity will not work in this setting because there are no similar sequences, right? So we train on the training set, and then we evaluate on validation set that is completely different from the proteins from our training set. And then we also test on a testing set that is also completely different from the validation and also in testing or training. So we specifically like optimize this for this task. So here the IS also, then here we like also incorporate the protein-protein interaction networks using the graph attention network. The idea is just to take, like, before passing it to the MLP here, to project it to the embedding space, we also use graph attention network, and that is based on protein-protein interactions to like collect features or combine to learn from the neighboring interacting proteins also. Okay, so because we believe that if the sequence is enough for molecular functions, but for like complex biological process, most of them is very difficult to predict just from single sequence. So you need several proteins in order to be able to predict some of the biological processes and maybe cellular components. So that's why we also incorporated graph attention networks here. Here are the results for molecular function ontology. Yeah, and so our interactions here did not help, and then the best performance was the simple model. This is our baseline sequence similarity-based split data. And of course, the performance is not as high as in the kappa results because these are very hard proteins, right? So in kappa, you like models, they get over 60 Fmax, but these proteins are like considered as uncharacterized. There are no similar proteins here. Then we also did for biological process. Here in the biological process,
sorry,
yeah.
one small question. Do you
Yes.
take for each sub ontology, do you take always different training set, or do you take same training and validation set for all three of them, and then just measure the prediction for each of the sub ontologies?
Yeah, that's a good question. I didn't mention that. We used their annotation and we generated three different sets. So like they're independent sets for training for molecular function, testing, validation. Also for biological process, it's a different set. Like, because the proteins, the annotations, there will be like more proteins with PP annotations, I think, but less proteins with molecular function annotations. So we split them in different, and then for each sub-ontology, we trained independent models.
Okay, thank you.
But we keep all the classes in our embedding model, okay? So our embedding model has all the knowledge in Go, so we didn't separate the axioms, okay?
Okay, thank you. So this embedding model is for all the ontologies, but then you
Yes.
just
Yes, then we
okay,
just
yeah,
do the
yeah, thank you.
evaluation, and then the training proteins specifically for molecular function, specifically for biological process. And also for biological process, we have here we have for biological process and cellular components, we have a model which uses molecular functions, okay? So molecular function annotations for some proteins, for example, they have molecular function annotations, but they are missing biological process annotations, right? So we try to use their molecular function annotations to predict biological process annotations, okay? And then we also use the first model and then predict molecular functions and then use these predicted molecular functions to predict biological process. So it's like a two-step. We first predict molecular functions, and then from molecular functions, we predict biological process using MF like predictions as features. And this gave actually like, I think the little bit improvement over like using simple ESM embeddings here, because it had some knowledge of molecular functions already learned, right? So this was the best model in the BP evaluation in our baseline comparison. And in cellular components, like our method that used molecular functions did not perform much as in biological process, but here we have the PPIs and ESM embedding based model giving the best performance. Okay, yeah. So, and then we had another second data set of uncharacterized proteins from NextProt database. So the NextProt database is developed by people from SIB, Swiss Institute of Bioinformatics, and they manually annotate proteins. So they call them predictions because they are using their literature, and I think their process is a bit different than I think what's usually the experimentally annotated proteins, but they do it manually and they review it. They do a lot of manual work and they only focus on human proteins, and they say like there are many uncharacterized proteins and we need to characterize them, and their goal is to characterize those uncharacterized proteins. Okay, so they say it's predictions, but their predictions are also manually reviewed, and then they do a lot of like each protein by protein they do research and they come with these annotations. They have NextProt database, so they provided us their latest at the time latest annotations, and there were not many proteins, I think a few hundred, more than 200 proteins, and we evaluated our method, and then we compared with other methods like SprofGo, Tail, and DeepGraphGo. And we have shown that also DeepGO-SE have a good prediction, but it's not that better than just using from MLP with ESM, for example. It's a little bit better anyway. In biological process, the improvement is a bit more than just MLP, but we are definitely better than SprofGo and Tail here. So it's just a different proteins, only human uncharacterized proteins. Okay. And we also like you don't find any similar proteins in these uncharacterized proteins, and we wanted to see like how are like what are the scores, and we like looked at several predictions that are made by DeepGO-SE. So we found, for example, for this protein called FAM151B, that this class, for example, have very high prediction score. So the model was quite confident that this protein has this function. And it was also like annotated in the predicted or annotated by people from SwissPro. So we looked at the structures predicted using AlphaFold, and then we looked at structural similarity to other proteins. So the most structurally similar protein to this FAM15B, I think it's not the most, but one of the like high scoring proteins was this lipophospholipase, which had this annotation already. So it's like based on they are not sequentially similar, but their structure is similar. And based on structure similarity, you could, for example, predict. So it like even if the sequence is not that similar, like these ESM embeddings, probably they have this structural information, and using them is helping our DeepGO-SE to predict those kind of annotations.
Maxat, here, both
Yeah.
proteins are human, or it's only the first one?
I think both are human here.
Okay,
Both are human.
thank you.
There are also, I think there are some in other organisms. They are also annotated to this function, and but when I looked at their annotations, they were not experimental. They were also, I think, predicted based on some sequence similarity, but they are structurally similar to this protein. Okay, this was the one that had some, I think, experimental evidence for this protein. So that was yeah, just a case study. So we did also some ablation study. We looked like what is the basic version of Go, and there's also a version of Go which is called Go Plus. It has a lot more axioms and a lot more knowledge, but since I think it's not EL, it's ALC, and reasoning is much more complex, and like I think it goes to exponential. You cannot easily reason here. That's why it's not used as in widely, but it has more knowledge. So if we use, I think in the best models, we use the Go class axioms because we learned from them, but we had to remove some of the axioms that were not in EL profile. So we tested without entailment and with entailment, and we see that in each component we have slight improvements, at least for molecular functions. For BPO, the axioms actually did not help much, and also for CCO, for example. But for molecular functions, it was more clear. So yes, that's it about the DeepGO-SE. We still we are continuing our work on function predictions. We're thinking about improving the current models and what to do next, and looking at our limitations. For example, we are using the CL embedding models, and it has some limitations. It cannot model all the relations, like many-to-many relations in the ontology, because it uses simple trans-E formulation for modeling the relations. And for example, this intersection axiom, where the intersection of two circles is not a circle, and then there's some inconsistencies happening there. But it was in another method we use, instead of circles, we use boxes to learn the embedding of ontology, EL box embeddings. That overcomes these limitations. We might also use this in the future versions. We, like in this DeepGO-SE, we still like use closed world assumptions. So all the unannotated proteins, we consider them as negatives. However, in ontologies, it's not true. And if it's not annotated, doesn't mean it's negative. It might not be just there yet. And in future releases and future annotations, it will appear, right? So that's another limitations. So here in DeepGO-SE model, we don't combine it with sequence similarity, but when we I think we on the Kappa 5 challenge in Kappa, it's important to also use some kind of sequence similarity, because the time-based splits usually have some of the proteins that are similar in the training. They share high similarity, and the annotations can be also learned from these similarity-based predictions. So we don't directly also learn from 3D structure-based predictions here. So that's in one direction. We are trying to also incorporate or directly learn, because sequence encodes for structure, and the structure defines the function of the protein, right? And we don't also combine, for example, literature-based predictions, which are kind of important also if you want to win Kappa, I think, in my opinion. Yeah, that's it. Thank you very much. And if you have any questions,
Thank you very
I'm
much.
happy to.
Have a great talk and beautiful work, very original, and just thank you very much again. So, colleagues, please, questions, comments, don't hesitate. There was a question in the chat, Maxat. So, when you mentioned that checking your model, you removed some proteins, but still model was able to predict the kind of zero shot.
Yes.
So when you say that you remove the protein, do I understand correctly that you just set the zero label instead of one label, or?
No, I removed the annotations of the proteins. So I took, for example, some of the classes, right? They were originally annotated, right? So usually what you do, if you have a class and you want to train a classifier, you keep some proteins in training that are annotated to this class, right? And some proteins in testing, right? So we completely took out some of the classes from the training, okay, that had some experimental annotations. We removed them completely from training. There were no instance of that was annotated to that class. And then we just used those in the testing set in a zero-shot manner. So that's what we call zero-shot. I think in some other cases, the zero-shot, it's a differently defined, but in our case, that's what we mean. So those Go classes without any training instance, we are able to predict because we learn an embedding for these Go classes using the ontology embedding method.
Okay, thank you very much. Colleagues, don't hesitate. Any other questions, comments? So, Valery, does it answer your question? Okay, so hope yes. Either please comment.
Yeah, thank you for the talk, Maxat. Very, very nice. And congratulations on your score in Kappa. My question is, this is kind of following a conversation I jumped in from another place about a paper we're writing that's trying to look at the problems of propagating function through paralogs, which might not be that great, versus orthologs, which may be okay. And my question is, are you looking at the type of homologs, at the type of sequence similarity that you're looking at? So for example, if you have two sequences that come from the same organism, usually function you should not propagate function through that. They're usually different functions. So what is known as the ortholog conjecture. Are you looking at that more fine-grained type of similarity, or you're just propagating through any similarity in function?
Yeah, we usually don't. We just use the similarity in any type. We don't separate. I mean, in my experience, but that's I think a good point. And yeah, propagating these similarities doesn't work all the time, because even like in other organisms, right, you have some similar proteins, but you don't have this function, right? They don't all, if they're similar, for example, you have some protein in rat and in human, they are 100% similar, but they don't share all the same functions, right? Because some of the functions simply is not there in that organism, for example. Or
okay. A second question, if I may, is regarding unseen labels. So supposedly you have, I don't know if you checked that, but you know, it's a classic case of function prediction that you have labels that are not in your training data, yet they may appear in your test data, especially in Kappa, where it gets it. So the labels exist in Go. It's not, they're not unknown, but they're not in the training data. Are you able to predict existing labels, labels that don't exist in the training data accurately if they appear in the test data, like in Kappa test data?
yes, that's what we do in zero shot, right? That's what we do in zero shot predictions.
Okay,
But
that sort of passed me. Okay, thanks.
yeah, that's what we do in zero shot predictions. But I think in Kappa this year I did not submit them, because I did not submit them because I think they were like, if I do this, I have to, like, for every protein I have to predict like 50,000 classes, make 50,000 predictions, right? And then in Kappa,
Well,
we
you
have
have you don't have
100,000.
to. That's a limit.
Yeah, that's the limit, right? I mean, if I want to predict for all the functions, and what I'm
Okay.
saying, it's like it was. So I think I just predicted those where I had some experimental proteins. But in DeepGO-0, we are able to do that, and we have shown that with AUC of 0.9, in some cases we can do that.
Thank you.
Thank you, Maxat. Here is a question. No, so it's something about quantum graphs. Well, I haven't heard what is it, but can you consider protein interaction networks as quantum graphs?
Yeah, I really don't know what quantum graphs are. I think I haven't worked with them much. So I'm sorry, I don't think I can answer this question.
Yeah, me too. I haven't heard this also. Okay. Colleagues, please, any other comments, suggestions, questions, whatever?
I have one question, which concerns that we learn these embeddings of the ontology. I mean, I think I kind of got the concept, but I'm not really sure how to train such a model to learn these embeddings. And my question would be if there is simple example in code which I can look after, and maybe you can send in the mailing list or something like that. Sorry, it's a little bit loud here because I'm in the train.
Yeah, sure. I think, I mean, all the code of EL embeddings and DeepGO-2 is available. You can look how we did this. So basically we just construct these loss functions, and then we use the axioms to generate these loss functions. We optimize them. It's very similar to what people in knowledge graph embeddings do, like trans-E. But here we have the difference that we represent concepts as n balls, not as points. And So the code is available. I can send everything is online. Code 2 is the repository. And if you have any questions, just create issues here.
Okay, yeah, thank you. I mean, I was thinking maybe there is a
yeah, I will be happy to answer.
I was thinking maybe there is this example with the family relations, and you know, that something like this, which is first as a first step to understand a little bit, you know, the basic concept behind this, because I don't know.
I think AL embeddings. Yeah, look at the EL embeddings, for example, that are the original code. So there we just use the PPIs and there's another, like, we even like developed this library called machine learning library with ontologies, and it has a lot more embedding methods, not just the EL embeddings, but there are many other EL embeddings here. So you can use this library to learn ontology embeddings.
Okay, thank you. Thank you.
There are some more questions in the chat. So, what was your training data?
So the experimentally annotated proteins from UniProt, I think there are many. I think there are around 80,000 proteins, right? And when you split the biological process and molecular functions, cellular components, the numbers go down to maybe 50, 60,000 in each subontology, something like this. And then we split it when using the similarity split. So the training and validation testing will be around 2,000-3,000 proteins, I think.
And so what hardware did you use for training, and how much time it took for?
Yeah, it takes maybe several hours. It's not that big, the model. And we use this ESM, we don't train it, we just get the embeddings and save them. We don't train, and to use ESM, you need, I think, at least 24 or 16 gigabytes of memory onto a GPU. We use I use mainly P6000 and A100 GPUs.
Okay, yeah, thank you. Colleagues, any other questions? So don't hesitate. Maxat, may I ask again about this zero-shot learning? So you already explained the idea, but maybe I haven't get it precisely. So what is the intuition? How does it work? So you do not have some annotation in the train, but still the model is able to deduce it. Just a small question to clarify my question. I mean, so do I understand correctly that we are talking about the annotation which is somewhere bottom at the child, yes? Because if it's annotation somewhere in between, then it's easy. If it has a child, then it must be one, yes? So it's not interesting, yeah? So we are talking about something like which is deeply down, yes? The last layer, yes? Okay, okay. So can you a little bit explain the intuition? How does it work?
So I think I can show you this, for example, this example here, right? So you have, for example, this class, Go class, called cell activation involved in immune response, right?
Yeah.
This is in the ontology. And this is equivalent to cell activation and part of some immune response, okay? So this class is more specific than this one. These two are more general, okay? So suppose you don't have any proteins that are annotated to this class. Okay. But you have some proteins annotated to this class, some proteins annotated to this class, so you basically can predict them. Okay. But this one don't have any annotations. But if you predict an annotation, a protein that has this one, and it's also has part of immune response, right? You can use this definition here and then infer that also this should be the annotation for this protein. Okay.
Yeah.
So this is done not in the manual way, but in the embedding space, okay? Because our embedding space preserves these relations between the classes, okay? So if you just, you don't do this, just use the embedding. We learn the embedding for this one, you learn embedding for this one, you learn embedding for this one. We train our model. Of course, these embeddings will be optimized using some protein embeddings, right? In this training process, but this embedding will be optimized only using the axioms in Go. This will be like learned based on the axioms, okay? And in the prediction process, now that you learned everything, now that you learned the embeddings for these classes, you can now use this embedding to do the prediction because you know protein embedding, you know the Go embedding, and you can like compute the prediction score for this class as well, even though you didn't show any training instance for this class.
Okay, thank you very much. So it's something like we have two circles and something which is just in between, in the intersection.
Yes.
We kind of infer from zero.
Infer from this, yes.
Yeah, that's great. Yes, yeah, very kind of impressive. Thank you. Yeah. Okay, thank you very much. So colleagues, please don't hesitate. There are many thanks in the chat. So another question. So what do you think are the most kind of, how to say, what are the pains of our clients, as business people say? So what biologists, have you heard something, what they want from this kind of system? Most kind of, some cases where they need to, maybe some urgent needs or something. I mean, something like that. I've heard some talk recently from the some PI from the Pasteur Institute in Paris, and she explained there is some research recent 5 years in immunology, and they found some similarity between bacteria proteins and eukaryote proteins, which were not seen before. And so they are kind of looking for these methods to, as you said, something like 3D space similarity or something like that. So, but maybe you have some other examples.
Well, I think in general, like predicting, if you can predict functions very well, it can be beneficial in many areas, like starting from drugs, like developing drugs, or to some industrial applications where you have some enzymes. And many people are looking for some specific proteins with some specific functions. And nowadays, even like there are ways to generate these proteins. Like you have a function and then you just generate the protein, right? So I think, I mean, this problem is very quite important, and it has applications in many places. But it's quite, in my mind, it's quite far from being solved now. But yeah, like with these latest developments in protein structure predictions now, we have, I think, very, like, the improvements are coming, and it's going very fast now. And I don't have any like specific applications, but I can like list many. I think it's quite important. I wouldn't say it's like very important in this, but it's also like there are many ways.
Another question. So my feeling when I looked at the data during CAFA, that the data is extremely noisy, that we have some proteins which are kind of, I mean, same. And for example, I look, for example, for some histones. It's just histone from, I don't know, cat and human. It should be many functions which are similar, but in the annotation, they are not that much similar. And I think this is just because kind of the data is noisy. So Gene Ontology is also people who are doing this, and so they cannot, they have some capacity. Yes, they cannot walk through all the literature. And so what's your feeling about that? That kind of, I mean, first, do you agree that the data is noisy? Second, what should we do about that? And the third, no, I think our methods should kind of help this. Yes, if we predict similar functions, then Gene Ontology team should somehow put this into the into their database. But I'm not sure that this is happening.
Yeah, it's yeah, I think, well, I think they don't, the reason they don't include these predictions in their database basically because they're predictions, and I think they have specific way of doing these annotations, some process, and until it comes to their, like, it meets their expectations, they won't hit. And it's quite noisy, yes, it's true. And these annotations are changing. You see some proteins they have at some point, and some of them they lose, right? Some of them they gain, some of them like, and it's incomplete. But yeah, I mean that's what it is, and I'm not sure, like, yeah, what we have to find a way to work with this noisy data.
Yeah.
And it's quite complex also, and I think that's what it makes a very interesting and challenging task to work on. And yeah, it's been like more than 8 years I'm working in this, more or less. So and it's still very interesting, not solved. And
Yeah, but for example, during the CASA, one of the participants, Zoltan, he proposed something like we should consider some subset for training, which is kind of contains most of the annotation. So his condition was something like we should look at the proteins which have annotation in all 3 subontologies. And when I tried it, for me, it boosted the performance. So the idea seems to be natural. We should choose training set which is mostly clean in some sense. So do you invest your efforts, your time in something like this? So now we have lots of proteins, but if you kind of maybe choose among them the most something best, or I don't know, something like this. So create your own very clean training set.
yeah, I agree, like, to so if you of course, if you filter out all the noise and then you learn only from those proteins that are well studied and they have many annotations, you are sure that I think your model will learn a very good model. You will have very good model, I think, but it will also miss a lot of knowledge as well, right? Because there are many proteins that are not well studied, and the way this evaluation is happening, it's time-based, right? So over time, it's not like you only on the annotations do not come like for the only very well studied proteins. Usually they come for the specific ones, right? If there's some similarity, structural, or if they did something in the literature. It's yeah, I don't have the correct and clear answer for these questions. I think it's matter of trying for. And usually, like, the CAFA winners are the methods that ensemble many different types of prediction. So if you want to win CAFA, I think you have to come up with many different ways of predicting functions and then some way combine. For us, like, our main focus is not only to predict functions, but also learn from the ontology. Like, our research questions are focused around the ontology knowledge. So yeah, I don't have like much, I didn't spend much effort into cleaning the data and looking at the protein annotations and this. So our focus is mostly the ontology part. That's what we work on as a group, as a biontology research group. And we think that, yeah, there are many other interesting methods that work, that have better results than what we do, but our focus is a bit different. So we don't
Okay.
spend in these things. Yes.
Yeah. Okay. Thank you. Thank you very much. Maybe a
Yeah.
little bit funny question. So, you know, this AlphaFold is a great success from Google DeepMind. So what do you think? Will they come to the next CAFA and create something like new AlphaFold which would completely solve the problem?
Well, they might, right? Well, who knows?
Why
People thought that protein structure is very far from being solved, and they were saying like they will like work on it for so many years. But I mean, now we have this AlphaFold, and nobody expected it, right? So who knows?
Okay, why didn't they come to CAFA
Yeah.
5?
Well, they have some their own maybe way
Okay.
of doing things also.
Yeah. Okay.
Thank you so much, Alexander. I think I have to go now.
Yeah. Okay. Thank you very much, Maxat.
yeah.
Great talk and great work, and I wish you success. And thank you very much again.
Same. Thank you for the invitation.
Yeah.
Yeah.
Okay. Colleagues.
Let's keep in touch, and
Yes.
yeah, if
Yes.
there are future CAFA in
Yeah.
Kaggle, we will
Yeah, we can
participate.
join the
Yeah.
teams. Okay.
Thank you so much.
Thank you. Thank you very much. Good evening, everybody. So video record will be, I hope, tomorrow in our channel, and I'll send by email and the link. So you will be able to see it on YouTube. Okay. Thank you very
Thank you.
much. Goodbye.
Bye bye.